Hello students, welcome to the biology class. Today we are going to continue with our chapter Biomolecule. Along with that, we are continuing our topic enzymes. Today's session we will start with the nomenclature of enzyme, means the naming of enzyme. Enzymes have been named by adding the suffix "ase" to the substrate on which they act. For example, the name of enzyme is sucrase. Ase is there and the substrate is sucrose. Similarly, we are having lipase where the substrate is lipids. So this is the one way of naming the enzyme that depends upon the substrate on which the enzyme is going to act. Number two, enzymes have also been named according to the physiological activity it catalyzes. For example, they are known as oxidase when they are oxidizing, dehydrogenase when the removal of hydrogen takes place, decarboxylase during the removal of carbon dioxide. Enzymes have been named according to the source from which they are obtained. For example, papain from papaya. Sometimes the name of enzyme gives no indication of the reaction or the substrate, for example, pepsin, trypsin, and so on. Next is the classification of enzymes. The International Union of Biochemists, that is IUB, has classified all the enzymes into following 6 classes depending upon their physiological activity: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So this is the flow chart in which the classification of enzymes are there: oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. So what is oxidoreductase? These enzymes catalyze the oxidation. What do you mean by oxidation? That is by adding the oxygen or removal of hydrogen or removal of electrons, or reduction by adding hydrogen or adding electrons to the substrate of a substance. So oxidoreductase, because they are carrying the oxidation and reduction. Example is cytochrome oxidase oxidizes cytochromes. Glycolate oxidizes glycolate. Transferase, as the name indicates, these enzymes catalyze the transfer of specific groups from one substrate to another. Example, glutamate pyruvate transaminase. Transfer an amino group from glutamate to pyruvate. Hydrolysis. These enzymes catalyze the breakdown of larger molecule into the smaller molecules with the addition of water. So amylase, hydrolysis amylose, that is your starch. So starch, as we know, that is a polysaccharides. So they are being hydrolyzed along with the water. Lyases. These enzymes catalyze the cleavage of specific covalent bonds and removal of specific groups without the use of water. Lyases means, that is, cleavage, means breaking. For example, histidine decarboxylase cleaves histidine into histamine and carbon dioxide. Isomerases. These enzymes catalyze the rearrangement of atoms in a molecule to form isomer. For example, phosphohexose isomerase converts glucose 6-phosphate into fructose 6-phosphate. The last is your ligase. So these enzymes catalyze covalent bonding of carbon oxygen, carbon sulfur between two substrates to form a large molecule, mostly involving utilization of energy by hydrolysis of ATP. Example is that is RUB means ribulose biphosphate carboxylase catalyzes the joining of ribulose biphosphate and carbon dioxide in photosynthetic carbon fixation. So we are having a 6 classes, that is oxidoreductase, transferase, hydrolase, isomerase, lyase and ligase. Next is very important, that is your mechanism of enzyme action, and the hypothesis which we will discuss here is lock and key theory. It is the traditional theory of enzyme working which is proposed by Fischer in 1894. So in this image you can see we are having the enzyme and they are having a specific sites. These are known as the active site in which the specific substrate will come and bound with each other. So the enzyme, it acts as a lock and they have a specific key in the form of a substrate where the boundation takes place, the complex will form, reaction will go on because the substrate is known as a reactant. Enzymes helps in catalyzing the substrate and with that they will form the product, but the enzyme that remains the same and free for the another action. So here the enzyme is the lock which can be opened with the specific key and the substrate is a specific key. Then the reaction takes place and then the resultant is the product and the enzyme as a catalyst is a free. So that's why it is known as lock and key mechanism. So the substance on which an enzyme acts is called substrate and the resultant as a product. All enzymes have specific three-dimensional configuration, a part called active site where the substrate binds. The active sites, they act as a lock into which the substrate fit like a key. The point where the substrate is bound on the active site is called as substrate binding site and it is specific for the specific substrate. Substrate binding causes the lowering of the activation energy and the reaction to proceed at the faster rate. During the state when the substrate is bound to the enzyme, a new structure of substrate is formed and it is called transition state structure. The active site of enzymes now in the close proximity of the substrate that breaks the chemical bonds of substrate and the enzyme product complex is formed. The enzyme releases the product of reaction and the free enzyme is ready to take up another molecule of the substrate. So in this we said that the enzymes, they are having a very specific three-dimensional configuration structures known as the active site where the specific substrate will bond, that is known as lock and key. The enzyme is lock and the substrate is key. The bounding takes place, the complex is formed, and then the resultant into the product with the free of the enzyme for the another molecule of a substrate. So our next lesson on the same, that is enzyme, that is on the factors affecting the enzyme, in which we will discuss about the effect of temperature, pH, concentration and chemicals. And with that we will finish our chapter. Till then, thank you. Have a nice day.